Good afternoon. I'm going to talk about quantitative chemomics that is a novel genomic selection platform which has potential to bring revolution into cannabis genomics and breed variety faster and cheaper. I'm Chief Science Officer for Destiny Bioscience. We're based in Edmonton, Alberta, Canada. We are bringing one of the largest center of excellence for cannabis genomics. This research center does cannabis breeding, genome sequencing, SNP genotyping, and the tissue culture clones will be provided to the growers to have better consistency as well as yield in their grow. We have four divisions in Destiny Bioscience. Destiny Bioscience does genomics breeding research, Destiny Healing, which is preclinical research division for cannabinoids, terpenes, taking into consideration human genetic variation. Destiny Innovations, that's research division for extraction, purification, formulation development to develop new and innovative formulations, medicines for treating specific disease condition in cannabis. Destiny Naturals, that's another division for developing skincare products using cannabinoids, terpenes, and flavonoids to treat specific skincare conditions as well as day-to-day use innovative products. The question comes why we need genomic selection or why we need these new modern technologies for cannabis. We know that cannabis has been used for last thousands of years, and since last 100 years there had been breeding efforts to breed cannabis and develop varieties. If we closely look at the history of cannabis breeding last century, we see that the most of the efforts were done to breed for THC. And recently we see some efforts. We went through thousands of strains and did meta-analysis, and I'm happy that recently there has been some interest in improving the CBD content in cannabis varieties, but most of the efforts were focused towards the THC-based breeding. And this happened because the technology was not available and the breeders were were dependent on just the impact of cannabis varieties when we smoke it. There was improvement in yield indoor because recently the other environmental factors which are important for cannabis yield, such as CO2 concentration and light and yield, water and nutrients. So all these aspects were kind of standardized, which improved the indoor yield, but we don't see much improvement in the outdoor yield because of the environmental factors such as heat, stress, and disease. All these aspects need genetic improvement, and there has not much work been done in that area. So overall, we see the century of cannabis breeding. There is a big scope to go into details of minor cannabinoids and understanding the genetics of minor cannabinoids as well as terpenes to develop better varieties and integrate all these aspects into the breeding program. So when we talk about genomic selection, it's very, very important to integrate so many physiological, genetic, as well as chemical components into it together to have that comprehensive breeding program, have that comprehensive integrated targeted breeding to achieve the goal of yield improvement on one side, but also keep the quality and consistency in the chemotypes that is we are targeting towards. Generally, in last 30, 40 years, we have talked about this much pound of cannabis that I grow, and that derives the price and that derives the profit. But I believe that it's the most important thing that should be considered is the active pharmaceutical ingredient that cannabis bud has, and that should be the deciding factor in terms of productivity as well as profit that we get from cannabis plant. When we talk about this integration, the breeding plant, the phenotype, chemotype, and genotype data, so all these aspects combined together decides the integrated breeding platform that one should use for the genomic selection. When we talk about what are the important yield contributing traits, then the pre-breeding that should be focused mostly on the traits such as how we can improve the root ratio, how we can improve the root biomass. When we talk about another important component that will push the yield forward is photosynthesis. How to improve the photosynthesis ability is one of the important key factors. So when we talk about yield improvement, all these sources which are carbohydrates which are providing sugars for cannabinoid terpene biosynthesis. These are the factors combined together to decide the bud yield, where the formula for the bud yield overall, we get the resource use. That's the resources that you provide. Resource use efficiency, how efficient the plant is to use these resources to end into high quality yield, as well as the chemotype. That's also one of the important factors. So all these factors combined together are very, very important when we talk about targeted breeding towards improving a specific chemotype or improving a specific trait. If we go into the details of the genomics of bud yield improvement, there are two factors. On one side, we can measure, we can see are the environmental factors. We talk about the stomatal conductance, we talk about the sunlight that's required, and the nutrients that we provide. We can measure, we can quantify, but on the other side, that's the complex part of the genomics of the yield, where we have to take into consider the expression of genes which are important in flower or reproductive tissue, the genes which are important for meristem maintenance, the genes which are important for root development. That's one of the holy grails in cannabis research, the root biomass, lateral root development, the genetics and genomics of it, the genes which are important for all these key traits which define yield in combination with the environmental factors. So that's the area that will push the cannabis breeding as well as genomics forward through the genomic selection breeding. If we see, there are three chemotypes now. We see in cannabis breeding mostly THC dominant intermediate varieties as well as CBD dominant varieties. But now we see that there are new products coming with many combination of high THC, low CBD, high CBD, low THC, and delta 8 THC, delta 10 THC, so THCV. So there are many, many more products are coming in market, and all these aspects as well as all these cannabinoids need to be considered when we select target for a specific cannabinoid breeding. If we look at cannabis plant, it's a very, very complex plant in genetics on one side because it's dioecious, and then it's heterozygous for the fullest expression of phenotype and chemotype. But on the other side, when we go into the details of cannabinoid biosynthesis, each cannabinoid is to be considered separately because it's a complex combination of many cannabinoids, many terpenes working together, expressed together to end into a complex phenotype and chemotype. For a simple example, if we talk about the total cannabinoid, we get a kind of normal distribution because we are talking about overall cannabinoid content. But if we look at the same trait from a THC inheritance point of view and CBD inheritance point of view, now there are two normal distribution curves. What I mean to say is when we go into the individual cannabinoid biosynthesis and genetics of it, it should be considered separately rather than traditionally it's considered as total cannabinoid that we are looking at. When we look at the cannabinoid biosynthesis pathway, there are transcription factors, there are acid synthesis which end up into the final cannabinoid that we can extract from the plant, but the overall biosynthesis pathway for each cannabinoid is to be considered separately when we are talking about the genomic selection model. In the Destiny Bioscience bioinformatics machine learning platform to establish genomic selection model, we have now identified most of the genes in flavonoid biosynthesis pathway. And we have also identified important genes in terpene and cannabinoid biosynthesis pathway from beginning up to the end. So these are the important factors for the genomic selection. Those are to be considered. We also went through genome-wide scanning and identified the important disease resistance genes in cannabis genome using comparative genomics approach. As I was mentioning, so all these key targeted genes combined together, we scanned through the cannabis genome, annotated it, and we have more than 4,000 key genes which are important for genomic selection panel. We have identified functionally, identified their annotated functions, and we know the location of these genes, and we are working on developing this first of its kind genomic selection panel, including the mathematical modeling that's required for the genomic selection. As this interesting graph shows, so we have annotated genome with a pan-genomics approach. We have key genes important for cannabinoid biosynthesis. We have key genes important for terpene biosynthesis. We have key genes important for flavonoid biosynthesis, their location, and as you can see in another graph, the other key traits such as flowering time genes, we have annotated all the genes and identified the SNPs in these genes, which will enable us to develop mathematical models as well as the panel for genomic selection. The resources that require for genomic selections needs the understanding of bioinformatics pipelines, using them effectively to annotate genes, develop SNP panels. But the real question is, why should we do it? What is the importance of using genomic selection? Because it really helps in terms of taking a whole genome approach at the sequence level and helps to access or helps to get back the missing heritability that we cannot get when we are doing phenotype-based breeding or when we are doing marker-assisted selection-based breeding, where the major genes are considered in terms of QTLs, but when we do genomic selection, when we use this approach, even the minor alleles or minor genes with smaller additive effects are considered, and this is one of the best methods that's the gold standard for cannabis breeding, I believe. And the other important aspect when we talk about genomic selection, that is, there are multiple traits, and we cannot consider each trait independently. We have to consider all these traits, ill-contributing traits, together, because in the end, we have to bring all these traits through breeding and genetics-based selection into the variety that we want to breed. And that is where the quantitative traits are important, for which the prediction models are built and the correlation coefficients to be added into all the quantitative traits, all the qualitative traits, all the agronomic traits which are important for the yield as well as biotic and abiotic stress. When we talk about combination of these traits, then again when we decide a targeted breeding approach, the models, the genetics, and the combination of the traits and genes we use changes depending on the targeted traits that is to be achieved. The key important aspect depends, the key important aspect which decides the correlation between what is the predicted phenotype and what is the expected phenotype depends on how big your population size is. It depends on the heritability of the trait, how many genes are, how many QTLs are controlling that trait, defines the success of genomic selection on individual basis as well as on the combination basis together. The other important aspect when we talk about the genomic selection and the real advantage we get from genomic selection is we can predict the phenotype before the just based on the genotype. And that's where it brings revolution into breeding, because we can discard so many, we can negate so many clones predicted from the genotype. And this is enabled when we build robust enough data sets of phenotype, chemotype, and genotype together, which are able to predict the phenotype of the next generation when you use this model. And that's where the predicted selection index goes on better as you combine traits and as you bring them together. Let's look at the three different methods that can be used and has been used. The first method, we know, traditional breeding method that has been used, and this traditional breeding method depends on phenotype and then chemotyping that's been done, crossing the plants and checking the pedigree for genetic improvement. And this has been the standard method that was used last 100 years for cannabis genetic improvement. The problem with this method, although I don't have doubt that was the only way and it has been successful, the problem with this method is that cannabis plant is very complex. Here we are selecting based on the visual traits. Here we are selecting based on the chemotype. But when we go into the details of the minor cannabinoids, when we go into the details of combination of cannabinoids, which can be really good for medicinal formulation development, we cannot do it because it has limitation in terms of understanding the genetics of it. It has limitation in terms of combining traits together efficiently. That's where last 100 years the breeding was happening. But recently, with availability of genome sequences and recent discovery of cannabinoid, terpene, biosynthesis genes, and the gene sequence available, we have moved a step forward. Now, we have molecular markers for these traits, and these molecular markers can be used for breeding and targeted trait improvement. But when we talk about cannabis, it's a complex plant, and all these biosynthesis pathways are complicated. And that's where, even if we use approach such as QTLs or such as genome-wide association studies, but still there is significant missing heritability, which is limiting us from using the maximum possible genetic gain that we can get. And that is where the genomic selection helps in terms of bringing all these different aspects of breeding together. We make use of all the high throughput phenotypic data. We make use of all the chemotype data and the genome estimated breeding value that comes out of genomic selection. That's one of the missing factor that really pushes forward in terms of getting that missing heritability brought back into the breeding program. But it also takes into consideration and uses the earlier available QTLs, which are major genes that has major impact on the trait, as well as the genome-wide association studies where we can select and use many QTLs together, many genomic regions which affect the trait together to bring all these aspects where we use the genome estimated breeding value, when we make use of the QTLs, and when we make use of the minor alleles identified through genome-wide association studies combined together to use this genomic selection panel. And that's where I believe the use of genomic selection panel is one of the important holy grails, or one of the important aspects those should be used for cannabis breeding. At Destiny Bioscience, using the pipelines for annotation, using the pipelines for SNP genotyping, using the pipelines for identifying the functional, putative functional traits, those are controlled by the genes. We have put together a very exciting platform and opportunity that will push forward and make amazing change the way cannabis breeding used to be done, and then we'll do the cannabis breeding at a very fast paced manner in a cost effective way. So to summarize, the cannabis breeding, which is complicated, now we have opportunity to make use of modern technologies, sequencing technologies, to integrate into the breeding, and that's going to play a major role in cannabis genetic improvement in the future. The maximum availability of the genetic diversity, that's a key resource. There is ample genetic diversity available through all these heterozygous genetic collections available, sequenced and annotated together for molecular marker development is going to be crucial. And again, it depends on, everybody has different collections, so the targeted breeding approach and the output that every breeding program will get depend on what is there available in their genetic resources in their bank for sequencing, phenotyping, and developing their marker development program. The role of high throughput phenotyping and chemotyping is going to play a major role because having these data sets properly collected, annotated, analyzed, and integrated into the breeding program is crucial for establishing these models. Chemotyping is also one of the key areas that should be integrated and should work hands in hand with phenotyping and genotyping, because having these databases and having access to minor cannabinoids, minor terpenes analyzed through in-house breeding program will be important to dissect traits separately and then bring that targeted breeding approach for focusing towards the minor cannabinoids and focus towards the terpenes, which were not considered earlier seriously, and the medicinal potential as well as the new opportunities that we can bring through all these minor cannabinoids. as I said, cannabis genome is very complex, so the genetics of these complex traits will be elucidated or will be discovered through this genomic selection platform, where high density genetic map and mapping populations, as well as the understanding of each cannabinoid biosynthesis gene pathways at individual level will definitely enable to even push the percentage of these minor cannabinoids higher, which was not possible earlier. There is a lot of scope to improve the concentration of minor cannabinoids through using this genomic selection platform. One of the really interesting opportunity that brings the research and discovery using these type of platforms is the untouched areas, such as improving the photosynthesis, improving the root biomass. So these are the areas actually which will push the yield forward. Why we see limitation in yield now? Because we are mostly focusing on improving the THC or CBD content, but we are not focusing to improve upon the basic yield contributing traits. Those are required for the yield as well as quality improvement. One of the key traits, such as improving the photosynthesis efficiency by selecting the genetic variation for light harvest complex, selecting the genetic variation for rubisco activase, and phenotyping for all these physiological traits is going to definitely help to improve the yield further, irrespective of the genetic variation that we are using as background. Another interesting area I believe we are focusing and should be focused on is root development. That's one of the key areas where the research and discovery is required for root biomass, lateral root development, root depth development, and genetics of all these traits. It's key to push all these yield contributing traits, as well as understanding the complexity of cannabinoid terpene biosynthesis through expression analysis, through genetic variation analysis, and through mutant analysis to bring that new wave of genomics-assisted breeding into cannabis research. At Destiny Bioscience, we are heavily focusing on data science and machine learning approach towards precision breeding, because in the end, we need platforms which can integrate all these data sets together and then predict the algorithms or predict the patterns which we can replicate again and again to add new traits into the breeding varieties. So to summarize, the genomic selection platform that we are developing at Destiny Bioscience will bring a big revolution into the cannabis breeding, targeted breeding, and opportunity to go deep into the discovery research of minor cannabinoids, enabling the extraction and purification of all these minor cannabinoids, combining together to develop better medicinal formulations. Thank you.
Hello, Christian Saucier. What are the additional efforts from the breeder's perspective when they engage with you or in your platform? What are the additional activities or processes involved, or costs maybe, for the breeder compared to more traditional breeding plan selection?
That's an interesting question. The simple answer is the maximum genetic diversity. More the genetic diversity, more the risk discovery, and stronger the genomic selection platform. So as more and more genetic diversity comes into the breeding program is sequenced and used for SNP genotyping as well as SNP development, stronger the genomic selection platform becomes. So adding more diversity, collaborating, and working together is one of the important aspects that we should do.
Great talk. So what are you doing in terms of instrumentation for your analysis? And then are you doing that analysis at different stages of growing to get that other paradigm in terms of what how maybe RNA is connecting to THCV levels at day 30 of the grow, for example?
Yeah, that's a very, very interesting question. In breeding program, we look at the genetic variation at genome level, but we also take into consideration the transcription, the gene expression, as well as different isoforms of the gene to identify like which is the functional copy and which are the minor copies in combination working for the traits. And yes, we take into consideration of expression of different genes at different developmental stage to identify the perfect expression or perfect stage which is responsible for the final chemotype.
Thank you.
One more quick question. So say there's a specific cultivar that the market or doctors want, and we know that they're really hard to find. Say like a THCV cultivar in NorCal that came out of Harborside or something like that, that's already been bred up to 13 or 14% THCV. How long will it take your sort of technology to create clones of that then farmers can grow?
That's a good question. So whatever these new rare cannabinoid strains are available, that's a great starting resource, and then we can improve these starting genetics for yield and other quality traits, or we can breed disease resistance into it, which is the real value to improve this variety and make it commercially successful in terms of growers getting benefit growing from it because the yield is optimized, there is disease resistance. So these traits can be combined together and bred into a new variety with our genomic selection approach and platform, I believe like within a couple of years. Thank you.